Thank you, Alvaro, for accepting our invitation to our Hive-side chat. We're very happy to have you here. If you don't mind, could you talk a little bit about yourself and then start your talk?
Sure. Good morning, everyone. Thank you so much for the Alberta Beekeepers Commission for the invitation. I am Álvaro de la Mora, and I am originally from Mexico. I started working with bees because when I was younger, still younger, Mexico was in the top 3 of honey producers in the world. So I wanted to be just a beekeeper. But I wanted to learn a bit more, so I studied veterinary medicine. And then in order to focus on honeybees. There, I did my first internship to the University of Guelph in 2011. Imagine how old I am. My main project in that time was working with Nosema serrani. Then I got the degree, started my master's in Mexico. I worked with Varroa, by Nosema, and neonicotinoids. Then in that time, I had the opportunity to work for a commercial beekeeper in Manitoba. That was in 2016. And in 2020, I started my PhD at the Honeybee Research Center. When I was in Mexico, I started my own beekeeping operation. It's still alive. My family is leading that. And right now, I am working as a postdoctoral fellow at the University of Saskatchewan with Dr. Sarah Wood. So this is a bit of myself. I will share my screen. Can everybody see my screen?
Yeah, can see your screen.
Okay, great. Okay. If for a reason you don't understand something that I say, or you have any questions, feel free to ask them in the chat. And if you want me to clarify something or to repeat something that was not clearly understood, feel free to do it. Today, I will talk about my research project during my PhD. This project was in collaboration with the Ontario Beekeepers Association, the Tech Transfer Program of the Ontario Beekeepers Association, and the Honeybee Research Center at the University of Guelph. This project was, well, my advisor was Dr. Ernesto Guzman, and this we focus on selecting or selective breeding for bees to low Varroa population growth. Let me just see if I can advance the slides. Okay. I am just figuring out. Okay, perfect. You can see my great. So first, I would like to start with some numbers. As we have seen in the last decade and more than 10 years ago, North America has had a higher number of colony losses. Just in the last winter, 2024-2025, Canada lost almost 40% of the colonies, and United States lost more than 50%. There are several factors associated with that, and one of them is Varroa destructor. As we can see in the graph in the left from CAPA, there is a lot of or high colony losses, above 30% usually, and this is unusual. Is not normal. The expected colony mortality was 15%. Now in the state, that threshold increased to 20%. So everything above that is not normal. And as I mentioned, the main responsible of this is Varroa destructor. Why Varroa destructor? Well, this is, most of you know, is an external parasite that feeds upon from the bee body. and affects the brood, as we can see in this image. Oops, sorry, okay, and adult bees. And in proportion to the size of a bee, the mite is really big. So this mite is a recent parasite that affects the western honeybee, Apis mellifera, which is the bee that we have in North America for beekeeping. This mite feeds upon the hemolymph, which is the blood of the bees, and the fat body tissue. The is equivalent to humans, irresponsible of metabolic processes and immune responses. This mite reduces the weight and protein levels on the affected bees. Is a vector of different viruses, for example, the four-wing virus. As we can see here in this picture in the bottom, the bee doesn't have well-developed wings, so these bees cannot fly, cannot protect their nest, cannot gather nectar, cannot thermoregulate their hive. This might also suppresses the immune responses of bees, kills between 20 and 30% of the brood, and shortens the lifespan of adult bees. And in a study conducted in Ontario, Varroa was the main responsible of colony losses in the province. Many beekeepers use synthetic compounds to control the mite infestation levels, but there are different disadvantages of using these products. One is that the mite can develop resistance against this product, and this has been well documented in Alberta, where these products are not killing a lot of mites as before. These products are toxic to the bees, and also they can contaminate honey and wax. And because of these reasons, there are some treatments that are not allowed anymore in Canada to use inside the hive. So there is a need of alternative solutions. One of them is the use of natural miticides, organic acids, essential oils. One of my current projects here at the University of Saskatchewan is assessing the efficacy and toxicity of oxalic acid in beehives during the summer and during the winter. Another alternative to the synthetic compounds is use biocontrol, entomopathogenic fungi, which are fungi that can kill the mite. However, these fungi can kill bees too, and the efficacy is not as high as using other products. In this picture in the right, we can see a pupae that is affected by fungus. And another alternative is breeding bees for resistance or for tolerance against the mite. So if we are talking about tolerance, we need to talk about honeybee immunity, how bees protect themselves against different pathogens. One of the we can divide the honeybee immunity into at the individual level and the social level. In the individual level, we have cellular immunity, which are cells, hemocytes. These cells are in the hemolymph, in the blood of the bee, and they protect themselves against different pathogens. The humoral immunity, we are talking about molecules, antimicrobial peptides, and different pathways and detoxification processes that protect the bees against pathogens as well, but also protect themselves against different pesticides, insecticides, etc. And in the social level, we can talk about behaviors. One of them is the hygienic behavior, where adult bees that are walking on the comb, they are able to identify cells where the mite is reproducing. Those bees open the cappings of the cells, remove the mite, or they just uncap and uncap those cells, and that opening and closing the cells interferes with the mite reproduction. The other behavior is grooming behavior. In this, bees use their legs and mandibles to remove the mite from their body and use the mandibles to kill it. And we can see here a picture in the right is a Varroa mite that has been mutilated or dented by the bees. What were the objectives of this project? Well, first, to develop honeybee populations with higher tolerance to the mite and lower levels of the deformed wing virus. We wanted to assess the health parameters on those bees, to identify the resistant mechanisms that those bees could have against the mite. We also assess the transcriptome. The transcriptome is a study of all the messenger RNA, which are sequences that codify to produce genes in the bees. And also we assess the metabolites. We quantify, well, we detected or we wanted to detect and quantify different metabolites present on bees. So we selected bees based on Varroa population growth. What we did, first, some of the results that I am showing today have been published. We have in total 5 publications. And one of the main goals of this project, we wanted to develop a methodology that is practical and feasible for beekeepers to implement in their operations. So the Honeybee Research Center has published videos on the methodology. The HBRC has different resources also, and the Tech Transfer Program of the Ontario Beekeepers Association has publications about this methodology. So what we did, we assessed the natural mite fall onto sticky boards. The sticky boards are these folder sheets coated with shortening or Crisco, the one that is used in baking, and they were placed in beneath a screen bottom board between the brood chamber and the bottom board. So we can collect all the mites on those screen bottom boards and quantify them. We left the screen bottom boards for 3 days, and to calculate the number of mites per day, we just divided the number of mites that were found on the sticky boards divided by the number of days that the sticky board was in the hive. And to identify the mite population growth, we assess the mite fall at 2 time points, one in the spring and a second one in late summer, 16 weeks difference. If the mite levels were higher in the summer compared with the spring, indicates that the mite population grew. So we selected colonies with the highest mite population growth, which I will call HBG, which is a susceptible genotype. And our goal was to select colonies for lowest mite population growth, LBG. Why we selected this for high population growth, high mite population growth? Because we wanted to compare our results. So after selecting those colonies, or identifying those colonies, we select the 3 most representative colonies, or the colony with the highest mite population growth, and the lowest colony with the lowest mite population growth. From those colonies, we rear 100 queens per genotype, and those queens were inserted into new hives. We had 100 hives. From those hives, we removed the original queen. We split those colonies into 2, and in each hive, we introduced one of the queens from each genotype. And this is in order to have similar infestation levels and similar conditions in the hive, and those colonies were kept in the same vineyard to avoid different or environment effects. Colonies were open mated. This is in order we selected only for queens, And this was in order, which this is to be easier for beekeepers to implement this methodology. I will present the results after 3 generations of selection. In total, we started with more than 300 colonies with different beekeepers, colonies that the tech transfer program was managed and was managing, and also the colonies had different genetic background. So we didn't care about if they were bugfast, if they were Carnolian or Italian. We were just focusing on the best colonies and the worst colonies for Varroa population growth. For our results, in terms of the Varroa population growth, we here in this graph, we can see in the horizontal axis the 3 generations, so 1, 2, and 3, and this is the fold change of number of mites comparing the mite fall on the summer compared to the spring. In green color, we have the colony selected for HBG, and in blue color, the colony selected for LBG. So the different literals indicates differences between genotypes and between generations. So see that after the first generation of selection, colonies selected for LBG in blue color have a significantly lower mite population growth, which was expected. When we continue the selection program after the different generation, each generation lasted 1 year. So we observed that there were no differences between the colony selected for HBG, the susceptible genotype, but we observed a significant reduction on the mite fall on B selected for LBG, comparing second versus the first generation, and we didn't see any difference between the generation 3 compared to generation 2, and this could be because we reached a plateau, so we couldn't go further down. When we assess the mite infestation levels on adult bees, we took a sample of adult bees, approximately 300 bees. We performed Varroa washes. Oops. Sorry, I am fighting with the mouse. Oops. And what we observed, these are the results after the third generation. So in blue color, here we can see the genotypes. Here we can see in blue color the LBGBs, in green color the HBGBs, and this is the percentage of infestation. And we can see a significant reduction on the number of mites on adult bees on the LBGBs compared to HBGBs. For the mite infestation levels in the brood, we cut a piece of comb with cap root. In the cap root is where the mite reproduces. We open it at least 100 cells, and we counted how many of those cells were infested. And the results were similar after 3 generations. Colonies selected for LBG in blue color have lower mite infestation levels compared with colonies selected for HBG. Then we wanted to assess the overwinter mortality. The overwinter mortality was assessed the following spring after the selection, after that generation was selected. And in here, in this graph, we can see the 3 generations. This is the percentage of mortality, how many colonies died. And at least in the first generation, we observed that we selected for LBG had lower mortality compared to HBGBs. We didn't see any significant difference between genotypes in any of the generations 2 or 3. However, this could be because of the management. Overall, the overwinter mortality that the Honeybee Research Center had in that year was really low, less than 50%. And as I mentioned at the beginning, every percentage of mortality higher than 20% now is not well, is not normal. So we were under that threshold. And this could be, as I mentioned, because of the management. But that happened at the colony level. But what happened at the individual level after we exposed the bees to the mites? So we had other set of colonies that were highly infested with mites. Those colonies were not related to the project. We collected those mites from those bees, and then we infested artificially bees from each genotype with the mites. So we ended with 4 treatments. 2 controls, HBG control, LVG control, which were bees that didn't have any mites, and bees that were infested with the mites. So we have HBG with Varroa and LVG with Varroa. And what we found, this is a survival graph, and we can see different 4 lines. Each line corresponds to one of the treatments. The first 2, black and red, are the controls. We didn't see any difference between bees that were not exposed to the mite. They survived the same. But what happened when we exposed the bees to the mite, the survivorship went down in both genotypes, indicating that yes, the mite is killing the bees. However, something that we were not expecting is that LVG bees here in blue color, after having the mite, they have higher survival compared to colonies selected for HBG bees. For a reason, these bees survive longer after they had or they were exposed to the mite. We also assess the foreign virus levels. We extract the RNA, quantitative in the left are controls, LVG control, HBG control, and in the right we have the Varroa treatments, LVG and HBG with the mites. And we are measuring the number of copies of the deformed wing virus. So if we compare only the controls, bees that were not exposed to the mite in cages, we observe that LVG bees have lower levels of the deformed wing virus compared to HBG bees control. But what happened when we expose the bees to the mite in both genotypes, compared within them, LVG bees with mite have higher mite infestation levels than the control, the same for HBG bees. And in both scenarios, bees with mites, bees without mites and bees with mites, LVG bees have lower deformed wing virus levels. So what are the significance of colonies have lower mite population growth? Adult bees have lower mite infestation levels, the same as the brood. Those bees have lower deformed wing virus. Bees that are infested by the mite have higher survivorship, and also the overwinter mortality was reduced at least in one generation, the first generation. Okay, now, does anyone have any questions, or we are okay to continue?
We don't have any questions in the chat,
okay.
So we can continue.
Great. Okay. Now, we wanted to see what are the possible mechanisms that are involved in the resistance of those bees against the mite, or the tolerance of those bees against Varroa. And we assess the 3 different behaviors, 3 different ways of immunity. behaviors, cellular and humoral. For assessing the hygienic behavior, we have a comb of a brood, and we freeze kill those the brood using a liquid nitrogen. Then we put the flame back into the hive to see how much the clean. So we have here two pictures in the for 20 hours, there were not too many cells that were cleaned by bees. And in the colony, in the picture in the bottom, we can see that most of the cells were clean. So what we found, here we have the three generations, first, second, and third, and this is the percentage of cells that were cleaned by bees. Again, in blue color, we have the LVGBs. In green color, we have the H. And at least and second, we observe that LVG have number clean cells, indicating that those between genotypes in the third generation, and this could be because first the year that we did that was 2021, 2022. That year was super dry, and the temperature and moist also affect in the mite levels, but also the mite infestation levels that we had, as I mentioned before, was really low. Could be that we didn't see differences in the third generation. We assess the grooming behavior on those at the colony level, and we look for the how many mites were mutilated. And in this graph, we can see again the three generations, and this is the percentage of mites that were mutilated. So at least in generation 1 and generation 3, colony selected for LVG of affected. We didn't see the difference in the second generation between the genotype, but the trend is the same. Assess the grooming behavior at the individual level. We did say for which also the time that the bee start grooming itself after we put the mite, and also the intensity. Intense groomer is because the bee moved more than two with and not so intense movements. However, working with mites is a bit challenging. So there was a another irritant. She used it with flour, the one that we use in the kitchen for baking. Then the bees have grooming instances after receiving irritant. So for the time to start grooming instances, in this graph, we have 4 bars, but the 2 in the left are bees classified as intense groomers, and in the right, bees classified as light groomers. If we start in the right, from the bees classified as light groomers, there were no differences. So bees that move less than 2 legs with not so intense movements, there were no differences between them. However, when we compare the bees that performed grooming instances in an intense way, LVG bees have higher reaction against the irritant. So indicating that more bees are performing grooming instances, and the bees that are performing intense grooming start faster. So they start grooming aggressive irritant. Stellar immunity by taking sample of the human and prepare a smear and looking at the hemocytes under the microscope. Higher the number of hemocytes, better the cellular immunity. In this graph, we see again 4 graphs. The 2 in the left are the controls, LVG, HVG. The 2 in the right are Varroa treated bees, LVG and HVG with Varroa. So for the bees that were not exposed to the mite inside the cages, so we can see the number of hemocytes, the number of cells. Bees that are not exposed to the mite had higher number of hemocytes. We were not expecting that. So for a reason, these bees have higher concentration. But when we expose the bees, when we expose the bees to the mite, Varroa suppresses the number of hemocytes. And this was expected, but it was not expected that LVGBs, after having the mite, didn't have a significant difference compared to control. And HVGBs, yes, they had a higher impact on the mite by reducing the number of hemocytes. Then when we assess the molecules, the genes related with immunity in those bees, we assess 2 different genes associated with immunity or producing antimicrobial peptides. So we have 2 different genes. Each gene are involved in a different immune pathway, defensin and hymenoptyricin. And we have, again, the 4 treatments in each of them, controls in the left, Varroa bees in the right. And for the controls, only in the defensin gene, we see that bees selected for LVG had higher expression of that gene. We didn't see any difference in the other gene between genotypes. But what happened when we put the mite on the bees? LVG bees express or increase expression of those genes, indicating that for the responses that bees have in this way is higher after they receive the mite. So maybe this is one of the reasons that those bees are not dying as much as HVG bees. So what is the significance of breeding in the resistance mechanisms? Bees have higher behavioral immunity by cleaning more cells with dead bees or dead brood, having more mutilated mites, and having more bees that are performing grooming instances. Also having higher numbers of hemocytes and higher expression of immune-related genes. There are any questions so far?
Does anyone have a question? Feel free to ask in the chat.
Okay. If not, we can continue. So
yeah,
far, we don't have
no
any
questions, but
okay.
Yeah.
Okay. Now we wanted to go deeper. What is inside the bee? What is happening inside the bee? So we assess the transcriptome, the transcriptome and metabolome. The transcriptome is the analysis of all the messenger RNA that codifies to produce proteins. So we assess all the genes and sequences that were expressed on those bees. And also the metabolome, what are the metabolites present on those bees too. So there is a publication from this project too. This project was focused on analyzing the transcriptome, the different genes that were present in the brains of bees that were doing grooming instances. This project was led by Dr. Nuria Morfin and was in collaboration with Purdue University. And what she reported, or was reported, was that there are different genes associated, but one of the genes that were highly expressed in the brain of the bees that were performing grooming instances after receiving the irritant were these two genes, odor binding proteins and gustatory receptor gene. This indicates that olfaction is a key part involved in the resistance mechanisms that bees have against the mite. And also, these genes are associated with hygienic behavior and grooming behavior. And these genes could be used or could be proposed as a potential candidate genes associated with Varroa resistance. So what we did in the same experimental settings that we had with the 4 different treatments of bees exposed to the mite within and bees not exposed to the mite. So we look at the genes that show differences between the treatments or between genotypes. In total, we identified 818 genes that were upregulated, and other number, less number, had were downregulated. In this figure or this graph is a mess. As a Corny says, graphs, right? The favorite graphs. So here you don't have to worry about identifying all of these genes. These are the different functions that the bees have, biological processes, cellular components, molecular functions, and pathways. In the left, we have the comparison of LVGBs with the mite versus LVGBs without the mite, and in the right, we have exactly the same, but for HBGBs. So for the number of differential expressed genes that were upregulated, there was a higher number of those genes on the HBGBs compared to LVGBs, indicating that HBGBs were more susceptible or responded higher or responded to the mite. Or LVGBs, in other words, LVGBs were less affected, if I can say that. Each of the lines represents the expression of those genes and the number of genes involved. So far, what happened what were those DEGs or differential expressed genes? Those genes were exactly the same that Nuria found, odor binding proteins, that those are associated with hygienic and grooming behaviors. Also, a higher expression of another gene that codifies for antimicrobial peptides, and others that are associated with energy production. And on the other hand, HBGVs, the susceptible genotype, after having the mite compared to their control, there was a upregulation of genes associated with detoxification processes and tissue repair. So this means that those bees, LVGBs, have higher responses against the mite, and HBGBs are more susceptible against those mites. Now, what happened in the unregulated DEGs? Exactly the same setting here, different differential expressions here. The comparison between the Varroa infested bees versus control for the LVGBs, and the same for the HBGBs. And for the number of differential expressed genes was higher in HBGBs compared to LVGBs. And what were those genes? Well, LVGBs have a downregulation of this gene, which is associated with nutritional stress, so less nutritional stress. And the HBGBs have higher expression of genes associated with energy and protein production. So those bees need to produce the production of the energy and proteins will be downregulated. What were the significance of this breeding program on the transcriptome of those bees? Well, LVGBs had higher protein production, higher energy production, higher detoxification processes, and higher tissue repair based on the function of each of the differentially expressed genes. Lastly, when we assess the metabolome, we are working on the manuscript of this paper. We had the 4 treatments. Each of the sample size indicates a replicate, a cage with this. So we extracted the metabolites, and then with this technique, we identified what are the metabolites that the bees have. So this is in order to identify and quantify those metabolites. In total, we identified 104 metabolites, and these are the different classifications: amino acids, fatty acids, organic acids, sugars, etc. And now you will see different set of graphs or columns, I will say. And each set of columns represents to one of the classifications of those metabolites. So we have amino acids, fatty acids, organic acids. Each of the groups have 4 graphs. 4 columns. The 2 in the left are the controls: HBG control, LVG control, and the 2 in the right are the Varroa, the bees with Varroa. HBGBs, LVGBs, the same in the right for fatty acids, organic acids. You will see different arrows. Well, each of the columns indicates one of the metabolites that we identified. And the arrows indicates when we compare Varroa treated bees, regardless the genotype, versus control, you will see where was higher. So the arrow indicates in which group was higher. Interestingly, and it was not expected, that Varroa treated bees have higher abundance of those metabolites, the majority. And we were not expecting that because the mite is feeding from the bee, so it's causing a nutritional stress, is taking resources from the bees. But when we were closer to see, okay, what is happening in each of the genotypes after they got the mite, the increase was because of the of the LVGBs. And what happened here? For those metabolites, amino acid or classifications, amino acids, fatty acids, organic acids, the majority had higher levels in the LVGVs. And for the other groups, sugars, nitrogenous compounds, alcohols, nucleosides, etc., there was exactly as a higher abundance or yes, abundance of those metabolites. So what were those metabolites? Those metabolites that got an increase was involved in immunity, neurological processes or responses, energy and muscular activity, and osmoregulation. So for the LVGVs, after having the mite at the metabolic associated with immunity, neurological responses, higher energy production, and muscular activity, and also they can osmoregulate better the impact of the mite. This was something that we didn't expect. So, what are the conclusions? First, that breeding bees for LVG or low Varroa growth is feasible and is practical for beekeepers. LVG bees were healthier because they had, or those bees had lower mite infestation levels and lower the foreign wing virus levels. They have higher hygienic behavior, higher grooming behavior, higher number of hemocytes, and higher expression of immune-related genes. In the transcriptome aspect, those bees have higher protein and energy production, lower immunosuppression, and lower carbohydrate or energy demands. And in the metabolome aspect, those bees have higher metabolites associated with immunity, energetic responses, and lower dehydration and hypoxia. But now, what is for beekeepers? Well, in Ontario, this methodology became mandatory for the bee breeders, so they can implement this methodology in their operations, and they can distribute queens among the beekeepers with queens that have a higher tolerance to Varroa. So this methodology is part of the Ontario Resistant Honeybee Selection Program, again in collaboration with the Tech Transfer Program, Ontario Beekeepers Association, and the Ontario Bee Breeders Association. And also in BC, honey or bee breeders are interested in implementing this methodology. Something important that I need to mention before to finish this talk is that for the result that I presented, everything looks amazing, right? Oh yeah, that is the key to go or to kind of fight Varroa. Some of the challenges that this project presented was first, the sample size. It's really hard for a bee breeder to implement this methodology, but because we have a higher samples, we need a higher number of colonies to select from. So if as an association of bee breeders, we work together, we can have a higher sample size in order to identify the best individuals. Another of the challenges that, not challenges, but some of the, I would say, some of the challenges that bees could have based on the results LVG bees could have based on the results that I presented in the transcriptome and the metabolome results. We saw that LVGVs have higher energy or expression of genes associated with energy, with protein production, the same as metabolites. So maybe having maybe those bees, in order to face the impact of Varroa, they have a higher energy and protein demand in order to compensate the impact of the mite. With this, I would like to end my presentation. And there behind this project, there are too many hands. And with this project, is because my name has a doctor in front of my name. So it was a great experience. I work with so many people, but part of my degree is because of them. And also thanks to all the scholarships that we received, awards, and also sponsors. Thanks.
Thank you so much. That was incredible. A lot of publications came up from that study. It's very impressive. So we don't have questions from our from people, but I have one question because I think everyone is thinking.
Yes.
How are the colonies doing? Do you like keep testing them or?
Well, one of the challenges also is the funding, right? So far, before I left from Wealth, it was 2000 end of 2024. We were just trying to keep the LVGBs, but right now, currently, we didn't have. Well, no, the Honeybee Research Center didn't have funding to continue with this project, but the main goal is to produce or to develop a methodology for bee breeders. So anybody can, or any association can implement this methodology on them. And asking your question will be something to do in the future, to continue this methodology for future generations.
Yeah. Okay, so Courtney, she has a question, but she's typing right now.
Okay.
But yeah. Another question that I have. Oh, there you go. I'll ask her first. Actually, Nathan was faster than Courtney. But yeah. Oh, there you go, Courtney. That was what I was going to ask as well. In thinking about initiatives for beekeepers, which brings me to queens. So yeah, she's probably gonna ask a question about queens.
Well, in that regard, we didn't want to, I mean, as the Honeybee Research Center, we didn't want to start. Okay. The Honeybee Research Center sells queens in only the Buckfast genotype or line, but strain. But we didn't want to sell queens in order to avoid any conflict of interests on the results. But the main goal is to, if you purchase queens from Ontario, yes, you can, those bees are selected for that. Thinking about initial incentives for beekeepers, which brings me, and maybe apply for more funding for bee breeders. I don't know if I answered the question properly.
Yeah, she was wondering if you had increased queen replacement in the HVG group.
Oh, sorry. Thanks for clarifying. I didn't measure that. However, something that one of the other challenges that we had that year, or the year that we were doing that, we had a lot of wildfires, not in Ontario, but the smoke was affecting the mating on those queens. And also, when we wanted to select drones and queens in order to identify effects on the drones, etc., it was really hard to get the mating and insemination. So I didn't I didn't measure the queens in that parameter. If I answer properly, I think so.
Yes, thank you. We have more questions. interest in the initial selection of HVG and LVG queens. I'm assuming that the selection was done following a treatment-free period. What, what was the initial timeline of monitoring for mites? How many samples? What was the cutoff level to qualify for LVG versus HVG by percentage of mite infestation? Sorry, lots of questions at once. I can repeat the questions if you like.
No, it's fine, thanks. I
Yeah.
can see also that question
Okay.
here.
Yeah.
So for the first question, for the treatment-free period, no, all the colonies were receiving the same management in terms of feeding, treating, etc. Because we didn't know if we could have promising results. So we treat in order to have this at the end. But maybe we can do that in the future. For the second question, the initial timeline of monitoring for mites, we monitor in the spring and in the late summer in order to see if we need to apply treatments. But for the second assessment in the summer, at least I mentioned 16 weeks difference, but at least 12 weeks will be enough. Also, the tech transfer program from Ontario, they presented or did a small research selecting bees on the mite fall, but also on the mite infestation levels on the alcohol washes. And they found significant results or similar results. And those bees selected for LVG had higher hygienic behavior compared to HVG bees. The cutoff level to quality to qualify for LVG and HVG, we just use a straight comparison. The colonies with the highest Varroa population growth were identified as high, and the lowest, and the colonies with the lowest mite population growth were identified as LVG. So we didn't have a cutoff there. We just selected the 3 worst and the 3 best. I don't know if I answered the question.
Yes. Well, thank you again. Do we have any more questions from the audience? Well, thank you again, Alvaro. That was great.